Good morning and welcome to Grand Rounds. I get the pleasure of introducing our speaker this morning. It's Dr. Anthony Samuel LaMantia. He's within our own Fralin Biomedical Research Institute, but a brief bio on him will let you know how accomplished. He got his Bachelor's of Science in Biology at the University of Chicago and went on to Yale University where he received a Ph.D. in neuroscience. From there, went to be a postdoc fellow at Washington University in St. Louis, and then moved on to North Carolina to become an assistant professor at Duke University. Kind of switched sides of blue and went to or joined sides of blue, an associate professor at University of North Carolina, subsequently rising to full professor. After these accomplishments, he moved on to be the founding director for the Institute of Neuroscience at George Washington University, being the Jeffrey A. Lieberman Professor of Neuroscience. In January 2020, he joined us here at the Fralin Biomedical Research Institute as professor and the head of the Laboratory of Developmental Disorders and Genetics. In July of 2020, he was appointed the director for the Center of Neurobiology Research there. He holds a joint professorship in biological science at the College of Science at Virginia Tech as well. Dr. LaMantia's research is always focused on the rules that guide how the brain is constructed and how and when those rules are broken to combinations of genetic mutations and environmental insults, individuals develop a broad range of neurodevelopmental disorders. His work has been supported for over 30 years by the National Institutes of Child Health and Human Development, the National Institute of Mental Health, the National Institute of Neurologic Disease and Stroke, as well as the Alfred P. Sloan Foundation, the National Down Syndrome Society, the March of Dimes, the National Alliance for Research on Schizophrenia and Affective Disorders, and the Simons Foundation of Autism Research Institute. He currently serves on the editorial boards of several leading journals, including Gene Expression, Developmental Neuroscience, and Synapse, and served for more than a decade as a senior reviewing editor for the Journal of Cerebral Cortex. He's been the senior editor and major contributing author to the textbook Neuroscience, soon to be in its 7th edition, the most widely adopted test for neuroscience education in the first year medical students, as well as advanced undergraduate students. As you can hear, he's quite well accomplished, and we're excited to hear a different take on neurodevelopmental disorders.
Thank you for that, Chris, and thank you all for the invitation. Thank you for getting up this early in the morning to be here, and to people on Zoom, thank you for listening to me in your pajamas. So it's always nicest to talk to your colleagues locally, because I think it gives you an opportunity to meet other people, and particularly because I came in January of 2020, and I can't remember something's happened right after. You know, it's actually nice to see people in 3 dimensions in Roanoke, because I was convinced that didn't exist for a while. So what I'm going to talk about today, as Dr. Pierce alluded to, is neurodevelopmental disorders. And I'm going to outline for you an approach of how to think about how these disorders that you see frequently in your clinical practice actually emerge. And of course, when you begin to talk about neurodevelopmental disorders, either clinically or in the research setting, there is an enormous amount of noise, and this wordle, which I love that word because it expresses what this is so clearly, it indicates some of that noise. I mean, you don't even want to read every word there, and what I want to do this morning is to strip away that noise and focus on the fundamental mechanisms that underlie pathology and how we might understand them. So we're going to strip away all of this noise, get rid of all of these things, and we're going to look at 2 things this morning. We're going to look at the idea of polygenic risk, the idea that most neurodevelopmental disorders do not have a monogenic origin. I think everybody knows that, but I think reminding all of ourselves that monogenic causes of any even particular aspect of a neurodevelopmental disorder are unlikely to explain most of the pathology and its consequences. The other thing is, another obvious thing, but I think important to remind ourselves of, is that neurodevelopmental disorders really are developmental disorders, that they reflect ultimately in their pathological foundations disrupted developmental processes. And in my mind, that sort of demystifies a lot of this, because even though the consequences sometimes don't even manifest themselves until much later in the lifespan, the origins reflect rational disruptions of cell biological and molecular mechanisms that we can identify. And if we know where the initial stumbles occur, it must be therefore possible to then figure out where the easiest point would be to intervene. And that's, I think, the hope of actually bearing down on the developmental disruptions, is to understand when the system diverges and thereby be able to think about ameliorating things. So, polygenic origins of neurodevelopmental disorders. Well, I think polygenicity, which is a lot of syllables, but a simple idea that multiple genes, if they are variants and they are combined, can cause some sort of pathology, has become all of the rage in genetics. This little cartoon shows that. Polygenicity is considered to be a risk predictor rather than an absolute predictor of disease. And I think more and more the genetic analyses of clinically diagnosed neurodevelopmental disorders anyway, like autistic spectrum disorder, ADHD, and even schizophrenia are bearing that out. So that there's not absolute prediction of disease, but there is an increased risk as you build a burden of genetic variants that are associated with disease. So, but risk for what? And the other question is, if this is genetic data, you then have to have a really good sense of what kind of genes. And obviously the risk that we're talking about this morning is a risk for neurodevelopmental disorders, behavioral disruption particularly that emerges over the early part of the lifespan. But as far as the kinds of genes, ultimately, they are all genes that are involved in the control of the development of not only the nervous system, but other organ systems as well. But of course, because approximately 80% of the genome is thrown at the problem of building a brain, it's not surprising that many of the genes that emerge as risk genes are indeed engaged in either the control of neural development or the regulation of neural function once a neural circuit is put together. So the kinds of genes are going to be exactly those kinds of genes, developmental control for the nervous system, and the risk is the risk for the emergence of these clinically defined behavioral disorders that actually can be, as you know far better than I, quite difficult and challenging for both the individual child and her or his family. So polygenicity is the basis for neurodevelopmental disorders. I think we are now all on board. So as of 2021, in a commentary written by my friend Dan Geschwind, he is a psychiatrist, but he stated bluntly, we have, like it or not, we have to understand it, and I think we could. And he made the point, and I think this is important because we're going to actually be looking at an example of polygenicity that begins with a copy number variant disruption of the genome. That copy number variants, as much as single nucleotide variants, SNVs, or single nucleotide polymorphisms, which are not as much mutations as they are just standard variation, if they're combined, they can actually give rise to different disorders in different individuals. And the same combination can actually, with a little bit of change in the genetic variants, actually result in somewhat different diagnoses, or at least somewhat different pictures of disease. So this is the complexity of what is now emerging as the reality of the interface between the genetics of understanding these disorders and what you see on the ground. And so I think responding to Dan's dictate and also to the reality of the situation, we do have to understand polygenicity. And the way that we're going to look at it is through the lens of a particular copy number variant disorder. Because copy number variants, which you know are either deletions or duplications of small to medium-sized or even large pieces of chromosomes, although the most survivable ones are smaller, are in a sense polygenicity in a box. There's a group of genes that are present in most cases in diminished or increased dosage. So in this case, stoichiometry is going to make a difference. But also, it gives you an opportunity to look across multiple genes when their function is disrupted at the level of the genome and ask what the consequences are. So we're going to look at this through the lens of copy number variants. And of course, copy number variants can be benign. We all carry them, or they can be pathogenic. And I think the one that we're going to focus on, you'll see as we go through, the polygenicity really does reflect, or I hope I can convince you, that it reflects the role of these genes in regulating very fundamental developmental processes that are used to build neural circuits as well as other tissues. So can we analyze this in a model copy number variant disorder as a starting point? So the copy number variant disorder that we're going to look at in detail is the 22q11.2 deletion syndrome, which I'll often refer to as 22q11DS. It's become a model polygenic genetic neurodevelopmental syndrome for an enormous amount of work clinically, both because of its association with schizophrenia later in the lifespan, and also because of the frequency of diagnoses of autistic spectrum disorder earlier in life, as well as a pervasive anxiety disorder that apparently these children face from their very earliest ages. So that's the neurodevelopmental part, but of course this is a syndrome. At birth, these children have congenital heart anomalies, which brings a lot of them to the attention of the clinical world because often there is a need for corrective surgery. This was one of the first genetic disorders that was fully characterized in the context of cardiovascular malformations. Limb and digit anomalies. There is a higher incidence of cleft palate. There's thymic hyperplasia, feeding difficulties. So this is a syndromic disorder affecting multiple organ systems, but you see that there is an enormous amount of phenotypic variation in the nervous system itself. When these kids get bigger, they have this group of behavioral disorders that are diagnosed clinically. They also have, there is some evidence of cellular brain pathology and cortical circuit dysfunction based on a handful of pathological studies and also an enormous number of functional imaging and live imaging studies. There are genomically accurate mouse models where we can begin to take apart the interface between the genetics of this disorder and the biology underlying the pathology. This is what we're going to focus on today. We're going to talk about mouse genetic models of this disorder and the contribution of individual genes to the phenotypes and the disrupted developmental mechanisms that underlie them. These models, we've shown, along with a handful of other labs, that the mouse models, including the model of the minimal critical deleted region, which I'll tell you about in a moment, is not only a genomically accurate, but a phenotypically accurate model. And I think I'll be able to show you what I mean by that over the course of the next several minutes. This mouse carries a deletion on chromosome 16 that is orthologous, has almost all of the genes that are contiguous in the deletion on chromosome 22 that causes the disorder. Now, mice don't get schizophrenia, mice don't get autism, mice don't get ADHD. If anybody tells you that they do, they're wrong. Send them to me, I'll set them straight. However, you can measure behaviors in mice that relate with some validity to those much more human-specific disruptions of behavior. And you can also analyze in detail the cellular pathology in the brain, the circuit dysfunction, and as you'll see, the developmental mechanisms that underlie these disruptions. So all of these clinically diagnosed disorders are considered to be disorders of cortical circuit development. We're going to spend a lot of time, therefore, thinking about how these disorders of cortical circuit development in the context of a specific beginning polygenic disruption emerge and disrupt cortical circuit organization and function. The other thing we're going to look at is the feeding difficulties and the underlying circuitry that actually is disrupted, leading to the perinatal dysphagia that's seen in these kids. So we're going to look at two separate examples of the development of neural circuits, and we're going to ask how this beginning genetic lesion disrupts them. So what is the genetic lesion? Now, you don't need to worry about the genes. When there's an individual gene that is going to be important for understanding the story, I will tell you what it is. There will be no quiz. There is an orthologous region to the minimal critical deleted region. This is the region 1.5 megabase deletion that causes all of the phenotypes that are associated with clinically with 22q11 deletion syndrome. The mouse has a contiguous region. 28 of the 32 genes are found there. The other 4 are not expressed in the brain, so we don't even worry about them. So this is a genomically accurate model of the human deletion, and that's really important because we are now dealing genomically with the same starting point to model the biology that we can study to understand the underlying mechanisms that are disrupted by this lesion. So we're going to look at this large deletion mouse. Remember, I'm a mouse. I don't get autistic spectrum disorder or schizophrenia. And we're going to use this genomically valid model as well as individual genes within the region that we can model to analyze developmental pathogenesis at high resolution. Okay, so now the puzzle of all of this is there are so many genes and so many phenotypes. I mean, I think you guys know better than I, particularly those of you who've seen these patients, that they are incredibly variable. The 22q11 deletion patient can have relatively high intellectual function, but have a number of craniofacial anomalies and other anomalies. Another could be very low intellectually functioning, have the cardiovascular defects. There are all sorts of mixes and variability within individual phenotypes. So, you know, this is a challenge, but we've decided that a key to solving this puzzle is to remember that this is a developmental disorder. So the way that you end up at the endpoints that you see reflects some history of development and the cell biological mechanisms that underlie all development that we can study rationally and begin to understand their relationship to the genetic lesion that starts this and how that leads to the phenotypic outcome. So what I want you to do is I want you to consider development as just a series of steps. It's a stairway leading to a landing where you actually get to roughly maturity or the finished organism. Now, of course, development goes on for a very long time, so there's no real final landing, but the metaphor will work for our purposes. So if we remember that development is polygenic and that it's also a series of steps, we can begin to actually ask, as development proceeds from fertilization onwards, what steps in particular might we want to look at in detail to understand where the stumbles are that lead to landing a little bit off-center on the landing for an individual with 22q11 deletion syndrome? How does typical development diverge, and where are the cell biological nodes that are actually compromised? So we're going to focus on the brain, of course, and we're going to remember that neural circuit development is a multi-step process. So like building any organ, you have to build the organ in stages based on generating the cells after the precursors have received instructions of who they're supposed to be, change their transcriptional program accordingly, then generated the cells, and then begin to put the circuits together. So we're going to look at those steps, the patterning of the early nervous system, how regions that are going to go on to make particular parts of the brain, even before they look anything like they're supposed to, actually have stamped upon them the molecular information that begins to give them the instructions to do what they're supposed to do. We're next going to look at the proliferation of cells, the actual division of the stem and progenitor cells that have to make the nerve cells that are going to make the neural circuits to underlie behavior, and how they begin to differentiate. And finally, we're going to look at, once you've taken those steps, the last steps of actually putting together multicellular arrangements that are called neural circuits, these groups of nerve cells that process information specifically. And we're going to ask whether or not there's a series of stumbles over each of these specific and analyzable cell biological mechanisms of development that actually elicit divergent circuits and suboptimal behavior in the context of the deletion of the genes that underlie 22q11 deletion syndrome. We're of course going to look at this in mouse, where we can take it apart in detail in cell biological and molecular detail. So what I'm going to argue is that there is a series of small stumbles over big steps during development, putting the right pieces in the right places, making the right cell types and the right numbers, and then knitting them all together for a neural circuit that actually underlies in a very rational way the pathology of 22q11 deletion syndrome, at least its neurodevelopmental disorders dimension. So there are deficits of behavior that we can relate very specifically to broader and far more complex deficits in humans. The first one is that the mice carrying the 22q11 deletion have a really hard time suckling, feeding, and swallowing. We devised a little mouse barium swallow test. They aspirate into their nasal pharynx. You can see that green stuff spreading in the deletion. It doesn't happen. They also aspirate into their lungs. They develop a pathology that looks like pneumonia. We prepared and cleaned up an antibody against mother mouse milk, and we found that these protein inclusions in the lung are actually milk protein that's been aspirated. So they're actually aspirating when they nurse. These get infiltrated with neutrophils, so that these animals actually have perinatal infections reflecting the dysphagia. So that they also don't gain weight as fast. There on the top is the weight gain curve for an infant with 22q11 deletion. That's male. The bottom is the mouse pup weight gain. You can see that the curves are similarly displaced, and the same thing happens for females. And we believed that it was worthwhile to look at this not as a craniofacial biomechanical disruption, although it is that to some extent, but rather that this reflected a disruption of neural circuitry, building the neural circuitry of the brainstem that starts out as this region of the hindbrain that's called the rhombencephalon, and it's divided into repeating units that will eventually give rise to cranial nerves that mediate suckling, feeding, and swallowing. We're going to go to the other end of things, and we actually identified a cognitive task that involves working memory, or as much as working memory as a mouse can have, which is the ability to hold online information to solve a problem in the future. If you want, Will. This is a reversal learning test. The mouse learns a rule that if he pokes his nose on a vertical versus horizontal bar, regardless of position, he gets a food reward at a distal location, and then he has to learn to reverse the rule. Mice carrying the 22q11 deletion are very bad at this. You can think of this as parallel to a lot of much more complex cognitive tasks that humans with 22q11 are disrupted on that have been measured. However, the most important thing about this is this reflects in very detailed physiological experiments that have been done a connection between one region of the association cortex in the mouse, the frontal association cortex, and the lateral and rhinal. So this connection, these cortical connections that are made by a particular class of cells that we'll talk about in a minute, are key for mediating that behavior. And we assume, and I'll show you, that it is indeed these connections that are disrupted selectively and specifically by 22q11 deletion when you build the neural circuits of the cortex. So we're now going to look step to step, and we're going to take one step at a time. We're going to look whether or not this patterning, the way that you change gene expression in local parts of the still very immature nervous system, but then predict what those cells can become based on beginning to change their local history of gene expression and thus their capacity to differentiate. So what we knew going into this is that there's a catalog of cardinal signaling factors. I won't list their names for you unless you need to know them. We knew that retinoic acid and sonic hedgehog were important for shaping the ventral part of the developing forebrain that gives rise to interneurons that are the inhibitory interneurons for the cortex. We also knew that there was a similar patterning event in the hindbrain that set up the front from the back and gave instructions for distinct classes of cells that make the cranial motor neurons as well as the cranial sensory neurons to do their jobs appropriately. So we asked whether or not this patterning was disrupted. We looked at whether or not signaling via retinoic acid, which is of course a metabolite of vitamin A, of retinol, was disrupted in both the cortex and the hindbrain in the large deletion animal. So what you see here is a marker of retinoid signaling in the hindbrain that marks the repeated units that are going to give rise to the cranial nerve at the back part, but not the front part. And what we're going to look at is what happens both in the forebrain and in the hindbrain to retinoic acid signaling. Here in the forebrain, you can see that in the ventral part, this is a reporter that tells us where retinoic acid signaling is happening based on turning on gene expression with a reporter construct. And we can see that there is a diminished level of retinoid signaling in the large deletion, and also the pattern has spread. So even though there's less, it's more widely distributed. There's also a change in the expression of a number of retinoid signaling genes that are actually regulated by the ligand itself. So there's a signaling change that begins to change the identity of what's on the cells, the progenitor cells that are in the ventral part of the forebrain that will then migrate out of there in some number and make the interneurons of the cortex. What we found by sorting those cells, we labeled them genetically, we sorted them, then we did RNA-seq to look at their transcriptional differences, large delta wild type. We found that there were a number of downstream genes that were changed, including the CXCR4 cytokine receptor. And what we were able to show is that there is a disrupted migration that actually flips the distribution of various classes of interneurons, thus disrupting the inhibitory circuitry of the cortex due to the diminished expression of the CXCR4 receptor because of the initial downstream, the initial genetic lesion associated with 22q11 deletion. So we can see that we're beginning to branch out in the genetic network and secondarily disrupt expression of genes that are not mutated, but by lowering their dosage due to this initial genetic lesion, we cause pathology. So that's the consequence of the altered retinoid patterning. Similar thing happens in the hindbrain. This is a wild type hindbrain. You can see retinoid signaling only happens in the back. In the large deletion, the signaling spreads all the way to the front part. Now, does that have any consequence? And are there genes in the region that might explain that? We had a sense that this particular gene called RAN binding protein 1, which is involved in the import and export of factors in and out of the nucleus, including that retinoic acid itself has to be shipped in and out, might be playing a role in this. So we knocked RAN BP1 out completely. So this is a single gene mutation. We see that we get the same change in pattern, but it's even more distinct. So what happens in terms of development of the circuitry? We can rescue that change in retinoid signaling by actually dropping the concentration of retinoic acid using another mutation that actually diminishes retinoid synthesis. And in the large DEL, carrying that mutation, we return the patterning to normal. So what happens? Well, there's the trigeminal nerve, which is derived from those anterior parts, those front parts of the hindbrain. It makes nice fascicles as the nerve grows out to the craniofacial primordia. This is what happens in the large deletion. The axons don't grow as much, their fasciculation is disrupted. This is what happens in the RAN-BP1 mutant. That phenotype of cranial nerve 5 is even worse. And we can rescue it completely. So we rescue the phenotype, not only the patterning, by diminishing retinoid signaling using the RLDH2 mutation that drops the signaling. So this lets us know that even at these early stages, when these patterning events diverge, that you're beginning to push the system in terms of its capacity to make the right neurons and to allow them to make the right connections toward a divergent fate that might influence function. So I've now shown you that the first step does give us a little stumble. So what about the next step, proliferation? Is there, when you're beginning to make from these progenitor cells that have now had stamped on them a normal pattern of gene expression that influences who they're going to be, and in the context of 22q11 deletion, an altered pattern, when you're actually making the neurons, do the stem cells behave differently? We're able to show in the peripheral sensory ganglia of the 22q11 deleted mouse, using tricks to label both the cells that are derived from the cranial placodes that make the mechanosensory neurons and those from the neural crest that make the nociceptive or pain-sensing neurons, that their distribution in the developing ganglia, and this is about mid-gestation in the mouse, so this is very early, changes so that the neighbor interactions change. So the cells aren't talking to each other properly. And you begin to make neurons earlier than you should. So you're actually depleting the population of progenitors, because what happens is that you get accelerated neurogenesis, and rather than having the precursors divide and giving you more precursors, they begin making neurons at an earlier time. And the assumption is, and we're now testing this, is that these earlier generated neurons become neurons without the right set of instructions to actually generate appropriate connections for the periphery, particularly nociceptive connections, so that we think that the actual pain thresholds during nursing are altered due to the 22q11 deletion. We're actually looking at that now. We actually looked at the underlying transcriptional variation. There are a lot of candidate genes, which I won't bore you with, but the other thing that we noticed, and this is something that is conceptually really intriguing, is that in any phenotypic domain, but particularly in this sort of domain, where we can actually measure things quantitatively with great accuracy, that there is higher variation. So what you're looking at is a plot of the coefficient of variation across all 18,000 genes that are expressed in the developing trigeminal ganglion. So this is one little part of the nervous system at mid-gestation in a wild-type animal versus a large deletion. And you can see the darker blue dots indicate where the coefficient of variation is greater in the large del, and the lighter blue are the wild type. 75% of the genes across the genome have greater variation in expression. So one of the things that we think that the 22q11 deletion is doing at the outset is it's destabilizing gene expression, quantitative levels of gene expression. So not only do you have a change in dosage of the deleted genes, you're beginning to have variations in the dosage of a whole set of downstream genes. And from individual to individual, it changes. So this actually is a way of beginning to rationalize the variability that we see. So we can also show that the same thing happens for the precursors of the cortical neurons that have to make these long-distance connections between association cortices that do the heavy lifting of cognition. And it's been shown in fMRI studies and in MRI studies that these association cortical regions and their connections are actually the target of functional pathology in 22q11 patients, and more broadly in individuals who've been diagnosed with these clinically defined neurodevelopmental disorders, autistic spectrum disorders, schizophrenia, et cetera. So if that is the case, and that reflects development, we should be able to identify divergences in the developmental program for making the cells that make those connections, which are these layer 2-3 projection neurons in the cortex. The upper layers of the cortex, they have long axons that project to other association cortical regions and make synapses. We're going to talk about how they're made very simply. There's an intermediate stem cell class called the basal progenitor that makes sure you get the right numbers of layer 2-3 projection neurons in association cortices. What we found is that these progenitors do not proliferate with as much, they're not as proliferative in the large deletion mouse, and you have fewer layer 2-3 projection neurons. And in data that I'm not going to show you, we looked at individual mice who performed variably on that cognitive task. We found that the fewer layer 2-3 projection neurons they make, the worse they were on that task. So that gave us a sense that the number of cells that you have and their capacity to make connections was important for the behavioral outcome. We also knew that several of the 22q11 genes were expressed in this region called the subventricular zone, where these basal progenitors that make the right numbers of layer 2-3 cells are found. So what we did was we actually identified these cells with a transcription factor marker called TBR2. We had a reporter that could label them green and fluorescent, and we could sort them in a cell sorter and actually do an RNA-seq experiment to begin to figure out, well, how do they diverge transcriptionally? We showed that the cells that we actually sorted expressed the appropriate markers for the subventricular zone, and to a lesser extent, the intermediate zone where newly born neurons are emerging before they migrate into the cortical plate. So this is an enriched basal progenitor preparation. This gives us more precise identification of transcriptome differences. What did we see? Well, we saw that the 22q11 genes are expressed robustly in these cells. And their expression goes down. So the bluer, cooler colors are down. Red is down in this block diagram. We also saw that a lot of other genes go up. And it turns out that there are 60 candidate genes that go up, 34 that go down. 12 of those are significant in there that are the 22q11 genes. And of these candidates, are they interesting? Well, you can do the standard sorts of analyses. Yes, they're involved in development and in CNS development. That's not as interesting, but you can begin to look at the individual genes. Now, those are all the 22q11 genes. They're going down by approximately 1-fold. But we picked out these candidates, and I'm just going to show you a little bit about why we think they're interesting. So, first of all, we know that the 22q11 genes are expressed in the subventricular zone, and we can show using a technique where those little red dots, those are individual messenger RNAs for a variety of messages. In this case, the message for CDC45-like, one of the 22q11 genes. We can show that the message in the cells of interest, both the basal progenitors, which are green, and the other cell classes, which are progenitors as well, that are blue, actually diminish by the same magnitude in the cells that is the case in the RNA-seq data. So we know that cell biologically we're changing the gene expression for 22q11 genes. But when we look at the other genes, that their expression also changes. One of the things that we noticed is that some of them have a very distinct expression pattern. They're expressed either in the anterior region of the cortical ventricular zone, where you're making progenitors, in the medial region, or the posterior region. And this gives us a sense that by disrupting gene expression regionally in these progenitors, you may actually be influencing the local neurogenesis of populations of neurons, particularly that contribute to the association cortices. So there is a distinct regional pattern. But more interestingly, we found that these 4 genes all go up, their expression changes, and why was that interesting to us? Well, it turns out that they are found in one of the most well-known imprinted loci in the genome, the DLK1 locus. There are several of these little stems with circles are regions of DNA methylation. If you're highly methylated, you don't express as much. If methylation goes down, you express more. So what we could do is we could actually isolate this region, and by bisulfite sequencing, look at the DNA methylation in the large del. You would predict that it goes down, and you can see that it does. So again, another displaced genetic change, genomic change. In this case, the deletion has caused a change in the methylation status of this particular region of the genome, changing quantitatively the expression of these genes. We don't know what this change, this increase in expression of these 4 transcripts means, and we're working on that. We do know that indeed these genes are expressed in the cells of interest, and they diminish in their expression as expected, including DLK1. We also know that DLK1 has been implicated in the selection to generate a postmitotic neuron versus remain as a dividing precursor based on earlier work that was done in ES cells that have been converted to neurons. So what we looked at was whether or not this is indeed expressed. It's expressed at low levels in the basal progenitors. But what we looked at was whether or not there is this premature neurogenesis going on in the large deletion. There are markers that we can use to measure this in the animal, and what we saw was that, yes, indeed, you are depleting the population of progenitors early by prematurely generating neurons when they shouldn't be generated. And we can do an assay where we actually look at why that happens. We can dissociate the basal progenitors. We can plate them at low density, and we can ask how they're dividing. If they're dividing and giving rise to neurons, we can recognize it with this red marker. If they're not giving rise to neurons but maintaining precursors, we can recognize it with the green marker. And if they're doing both, there's one red and one green. And what we saw was that you increase the red and the green, so you're depleting the progenitor population by kicking off a neuron earlier rather than making two precursors and keeping that population up. And that we think that this is what is actually cell biologically underlying the diminished output of layer 2-3 projection neurons. So now I've shown you that there are two stumbles, and now we're going to talk a little bit about the last step, the circuit formation. Now, we're going to look at this, what happens to the circuitry and how it regulates function in feeding and swallowing briefly. We did this by looking at the motor neurons that control the protrusion and retraction of the tongue. This is very important. There's a sinusoidal protrusion and retraction to begin the stream of milk that is then propelled through the oropharynx, past the slightly open larynx, and into the esophagus. If that doesn't happen correctly, you get altered swallowing. So rhythmic tongue protrusion is essential. We could identify by retrogradely labeling the green cells that project to the retractors and the red cells that project to the protruders, and we could actually measure physiologically their patterns of activity. So this is a protruder motor neuron. You see in the wild type, it's normally firing in rhythm. That rhythm is disrupted and it's not firing at all in the large deletion. And when we look at the retractor, so the protruders slow down. When we look at the retractors, the motor neurons that regulate them speed up. So we now have a physiological dysregulation reflecting this disrupted development of the brainstem that actually results in a change in the pattern of activity that regulates protrusion and retraction. And we can actually go on to show that this reflects a change in the GABAergic inhibition, the size of the GABAergic iPSCs and their frequency, and that this actually reflects a cell biological change. So while we're looking at the little red dots, which we've quantified with a color-coded pattern in the bottom picture, are actually individual GABAergic synapses. This is what happens to the GABAergic synapses in the large deletion. There are not only fewer of them, but we could show by electron microscopic molecular analysis that there's actually less GABA in each synapse. So there is diminished GABAergic synapse formation, and that results in diminished GABAergic tone to regulate the appropriate pattern of firing. So what about the cortical circuits? Well, I've told you that there is this deficit in this cognitive behavior that underlies those association cortical-cortical is underlined by those cortical connections. I've told you that if you make fewer of the neurons in the large deletion that actually mediate those connections, that the fewer neurons you have, the more likely you are to be one of these large cell animals that behaves, who can't do the behavior really spectacularly badly. So are these connections, once you've made the nerve cells, do they get what's left? Do they connect up properly? Do they actually compensate in some way? Or do they actually fail to make appropriate connections? So you have two hits. One, you don't have enough of them, and then they're not connected properly. And the answer to that is Yes, and I'm going to show you that that reflects a disruption of process growth and synaptogenesis due to the activity of one of 6 mitochondrial genes in the region. So one of the most common classes of genes in this region are genes that are expressed, somatic genes that are expressed in the mitochondria and regulate mitochondrial metabolism. And so what we found is that there is indeed quantitative underconnectivity, and the data here where you see black bars, those are distal cortical connections that there are fewer of them based on tracing experiments that were done quantitatively. So there are fewer cortical connections, particularly to that lateral entorhinal cortex from the frontal cortex that is so important for this particular behavior that we're looking at. The next thing we did was we asked whether or not these particular layer 2-3 projection neurons actually differentiated properly. Do they grow their dendrites in enough extent so that they can accommodate the synapses that have to be made onto them? We did what we call a genetic Golgi. We used genetically engineered mice where we could actually label selectively just a few in each animal layer 2-3 projection neurons in the frontal cortex. And what we saw, you can see this visually, is that the dendrites don't grow properly, they don't branch appropriately, so that you actually have a disruption of the territory onto which synapses can be made quantitatively. So does that change the number of synapses? The answer is yes. Also, when we looked at these cells at the electron microscopic level, they are under oxidative stress. They have all the hallmarks of oxidative stress. We measured this cell biologically, and then we began to ask why. We also showed that they really are synaptically disconnected. There are fewer synapses, and the synapses themselves are dysmorphic. They have fewer synaptic vesicles. So this is a circuit that not only are there not enough neurons in the circuit, they don't differentiate properly to make connections. So why is this happening? Well, we knew that there were these 6 mitochondrial genes. We actually knocked each of them down individually in layer 2-3 projection neurons in culture to see whether or not any of them disrupted dendritic growth and axon growth. Only one did, the thioredoxin reductase 2, which is a mitochondrial matrix enzyme that's involved in the clearance of reactive oxygen species. We could show that reactive oxygen species in mitochondria in these layer 2-3 projection neurons, as well as in the cytoplasm, are elevated. This gene is expressed in the mitochondria of layer 2-3 cells, both in vitro and I'll show you in a moment in vivo. When we actually enlarge deletion cells in this culture, so full 22q11 deletion, they have the same dendritic and axonal growth failure. When we re-express TXNRD2 at a normal level, we rescue that growth. We also rescue the elevated ROS. We diminish ROS back down to wild type levels. So this one gene is regulating oxidative stress through the metabolism of reactive oxygen species. This gene is actually involved in the clearance of reactive oxygen species by providing reducing equivalents for the PRX pathway to eliminate hydrogen peroxide in the mitochondrial matrix. So does this gene alone cause the same change in cortical neuron development? Well, what we can do is in a wild type mouse where all the other layer 2-3 projection neurons are fine, we can take one copy only out of one of their neighbors and ask whether or not it has the same effect on the development of the dendrites of these cells. And the answer is yes. The pink bars are what happened to the large deletion layer 2-3 cells. The green bars are what's happened to these individual neurons that are in a wild type animal that no longer have both copies of TXNRD2. So this implies that at this point in development, diminished dosage of this gene puts these cells under oxidative stress and disrupts their capacity to make connections. And this is just showing that the mitochondrial distribution and function is indeed disrupted. But can you rescue this? In vitro you can, because if we give metabolic feedstock through pyruvate or reducing equivalents through N-acetylcysteine, we rescue the ROS phenotype, we rescue the growth phenotype. We also rescue ATP production measured in real time based on oxygen consumption. So, but Does it actually do anything in vivo? And this is the answer. If we give the mice from birth onward N-acetylcysteine, first via maternal milk, because we feed it to the mothers, we can show biochemically that it gets to the pups, and then we put it in their drinking water throughout life, we rescue the dendritic phenotype. This is shown quantitatively here. We also rescue those synaptic changes, and this was really remarkable. This surprised even me, and I'm surprised at nothing. The synaptic architecture returns to wild-type levels. The frequency of synapses, their vesicular loading, their morphology. But most importantly, we actually rescue the behavioral deficit. So by basically, during the period, you don't have enough of these cells, but by restoring the metabolic balance in terms of ROS clearance, pharmacologically, we were able to actually fix the growth of the cells that are still there. We were able to fix the synaptogenesis, and we were able to fix the behavioral deficit. And we're now trying to figure out whether or not how all of those fixes are actually contributing to building a circuit that, even though you don't have the right numbers of cells, and even though, as I told you earlier, the interneurons that are saying no are not in their right places, you can still get enough plasticity to give you a circuit that can actually restore some behavioral function. So what have I told you? I've told you that this polygenic disruption for building cortical circuitry not only acts in terms of controlling the numbers of cortical cells, but also how they finally form circuits that mediate specific cognitive functions. And for a little good news, this is of course in mice. So, whether or not this is actually could be a foundation of a therapy in humans, I have no idea. That's beyond my pay grade. But at least we've shown proof of concept. So where are we? I've shown you that as you climb the stairway of development, that there are these cell biological mechanisms that are rational, that we can identify, and that these are actually the places where this polygenic disruption, in this case, this copy number variant, this deletion of minimally 32 genes on chromosome 22 in human, 28 in the mouse that we can study as a genetic model, actually does disrupt the cell biological mechanisms of patterning, telling the early progenitors, okay, you're in this part of the nervous system, you're supposed to become X, Y, or Z. That's the first little stumble. Then when they actually have to make the neurons that they're supposed to make, they don't have the right instructions, they don't make enough of them, they make them at the wrong times. That includes bringing different chromatin organization, the DNA methylation, for example, that actually may underlie this divergence of differentiation. And finally, a final stumble in putting together the circuits, the connections, the patterns of activity that have to operate behavior. I've shown you that those actually are divergent as well. So all of these stumbles do indeed add up to the divergent circuits and presumably the suboptimal behavior that those circuits underlie in this genomically accurate model of this polygenic developmental disorder. So small stumbles do indeed add up. I would argue that this is an important, essential dimension of understanding the underlying pathology of neurodevelopmental disorders, that it has to be understood through the lens of polygenicity, and that it does so not by just taking a big hammer to one single phenotypic domain, but by actually destabilizing quantitatively multiple regulated developmental mechanisms that lead you to this divergent climb up the stairs of development. So, polygenic risk, what's the score? Well, what kind of genes? Genes with multiple functions, including a variety of functions in developing neural precursors, stem cells, differentiating neurons. Risk for what? Risk for altered developmental cell biological mechanisms. So we're demystifying this. It's not that something bizarre has happened to the brain. You just haven't built it right using the cell biological tools that are mediated by specific genes during development. And so I want to leave you with the idea that neurodevelopmental disorders equal small changes in basic developmental mechanisms, reflecting, in this case, the altered dosage of genes, But you could imagine that any, even clinically diagnosed developmental disorder, neurodevelopmental disorder, could reflect, in a sense, a private mosaic of polygenicity, multiple variants that come together to give you a divergent outcome that in some way disrupts specific domains of behavior. And with that, I will thank the people who did the work. This is the lab in Roanoke. Even some people have masks. I didn't have a beard at the time when that picture was taken. In particular, Tom Maynard and Dan Meakin, who are faculty members within the lab and who moved with me from DC, were critical in putting all of this together. These people also did work, including Abra Roberts, who wants to do a pediatric residency, and she was my first VT medical student in my lab, and she was spectacular. Carney Flynn, who is also a medical student working on the feeding and swallowing stuff. All of these people did a lot of important things to make this work happen, and these are the people who paid for it, so I'm happy that they're there too. And with that, I will take questions.
Absolutely fascinating. The amount of work that went into that is incredible, mind-boggling. My biggest frustration, though, with how incredible all that research is applying it to the bench. Applying it to from the bench to the clinic. So, I mean, are you talking with anyone? Have you heard of anyone who's doing any clinical trials with N-acetylcysteine?
At this point, no. I mean, one of the things was that we published that paper right as we were moving. And my hope was that when I landed here, that I could begin to talk to people about exactly this. And of course, communication was very difficult because of the timing. There are a number of people who are interested in this at the 22q11 meeting this past summer. Several people talked to me and asked me about, if you were going to do a clinical trial, what sort of outcome measures would I suggest? This is not my expertise, but I think it would be really useful for people who can think about this, because this is one instance where there's clearly something that you can do and with verifiable, at least postnatal developmental outcomes. So, to me, the good news is also that you're not doing something prenatally, which to me would be a nightmare. But this is actually something that's relatively safe. N-acetylcysteine is used as a pharmacological treatment in I think, for some kidney disorders, if I remember correctly. Among other things, for oxidative stress, basically. There is anecdotal evidence and a few, they're not even, I wouldn't even call them clinical trials as much as they're case reports of groups of either children with autistic spectrum disorder or young adults with schizophrenia who have been treated with a variety of antioxidants, and this relieves symptoms. But I think this provides a much more rational opportunity to actually say, okay, and you can measure the outcomes. So I would be delighted if someone, I'm a molecular biologist and geneticist and neuroscientist, so, the translation to me is not something I'm familiar with, but I would be delighted if someone wanted to, at least we could advise. And and this is important. Actually, yesterday I was teaching the graduate students, and we covered the ASO therapies for SMA. And part of what drove that was that there was a genetically accurate mouse model that both the ASO therapy and the gene therapy could actually be investigated. You could look at the outcomes, the cellular outcomes in detail, and you could have confidence that there was actually target specificity in applying. So we have a parallel thing here for these cognitive circuits. But my hope is that we can, with people who know what they're doing, we can move forward. There are some folks at CHOP who have a big population of these kids who are interested in this.
A different lens, I don't know if this is a question, but more common, a different lens is, how do you bring it into the clinical setting? Although I look back, I told you previously that I come to this world as a country pediatrician, and the approach was solely the idea of how do we intervene, how do we make it better for the child. And that's still good, and that's important, but I think it's really intriguing when I look at this, that you take this where we thought of as a continuum that just happens into these little parts. It goes back to my world of engineering before medicine and looking at little control circuits. And it's kind of like if your cell phone has a bunch of apps, how do they all interact? How do you break that control loop? And you can intervene at any given level is what you're teaching us. How do we do that now?
Right. Well, and,
No, but you know, that's where we are now. Where are we going with this?
well, and I think you're bringing up a very good point, and it's one of the things that inspired me. Even though I call myself a neuroscientist, don't let this get around. My entire career has been inspired by cancer biology, because the cancer biologists got the right message. The way that you take the pathology apart is, first of all, you don't assume that it's all one thing. And then you begin to look at networks of molecular change. And so to me, and those networks of molecular change in cancer biology, what's the basic science foundation of cancer biology? Developmental biology and genetics. So when I set out to do this, I decided that I was going to bring that lens to looking at it rather than exactly for those reasons, because I think that the engineering approach of thinking about components, how they interact, and actually having a quantitative metric of that begins to give you more insight into the complex outcomes that you're looking at based on my understanding of these disorders. And it really is, I'm a basic scientist, and there's something beautiful about just taking biology apart. But, I was also trained by people who had brought genetics to the table in terms of understanding neural development, and I realized that the best way to do that was to use the human genetics that disrupts the development of the brain, but also in a way that could actually inform you guys as you deal with the clinical realities of these issues. So I hope that that's what we're doing. I mean, I think, we work in mice, we work at a very reduced level, this idea of looking at these molecular networks, because that's what's brought progress in the domain of cancer biology, and that may be the place where neuroscience, which, is sometimes resistant to these sorts of molecular analyses. Yeah.
Well, you mentioned that potentially your mouse model has more pain with feeding, is that correct?
Yeah, and we're looking at that now.
Have you tried treating the pain and seeing if the feeding improves?
You're reading our minds. So there is anecdotal evidence in the literature that treatment with capsaicin can relieve some aspects of perinatal dysphagia. Capsaicin, of course, is the ligand that binds to the TRPV1 channel, for which David Julius won the Nobel Prize last year. David's such a nice guy. And so if that is the case, there actually may be, and to me, this is at least a hypothesis that makes a little bit of sense. If you are a nursing infant and there is pain involved in the transit of milk, what are you going to do? You're going to be averse to the condition and not want to do it, right? And so we are actually looking at this. We are using metabolic mapping of intermediate early gene expression to actually look at the nociceptive sensory relay neurons and their activation versus the mechanoreceptive. We have ways of molecularly tagging them. So we're taking it apart like that. But the idea of, this has been looked at in humans, and apparently capsaicin periorally, so it's not given systemically, it's actually ruled. Which sounds awful to me, but I guess, if it works. But what would be the rational basis? And this was a way of looking at it. So, other questions? Well, thanks for the opportunity to meet you all and come and tell you the story. It's always fun to talk to a new audience. And I am on the Riverside 2 campus on the second floor of Building 2. If any of you have any questions, if there's any information that you think I could provide for you, and whatever you're doing, please don't hesitate to email, call, or just show up. So thank you all.
Thank you.